So this is the topic now. I think Dr. Sunil Gupta showed us that in spite of having such advancement, we have, I think, 7 companies promoting insulin. Why not 40 companies? GLP-1, 144 companies have filed. And how is it possible? And how do we choose between them? Now, I'll take you through, I can't see here anyways. I'll take you through a very simple thing. Whenever the several companies comes to you and tells you that we have now this biosimilar in GLP-1, you start writing it, ask them a few questions. The first question is that none of the regulatory authorities, whether it's India, CDSCO or FDA or EMA, requires that your HbA1c or your weight reduction should be matched. That is not mandatory. You know, I can get 90 out of 100 in class and Jayant can get 90 out of 100 in class. Doesn't make us the same. We might get the same marks. So you might show that both have equivalent reduction in blood sugar. Doesn't mean that the two molecules are same. All the regulatory authorities want is the molecular structure is the same, number one. And number two, the absorption in the system is the same. Good enough. Nothing else is required. And these are the only two data where none of the companies are willing to share with us. For whatever reasons, we don't know. Now, another thing, since a lot of the parent companies will come and tell you that process is everything. How we make the molecule is everything. Like insulin used to be made from E. coli. The biosimilar Biocon made it from yeast, but the end product was absolutely the same. The absorption was same. So both of them have EMA, FDA, and Indian drug controller approval. Here, suddenly the controversy has come that biological way, through yeast, the parent molecule of Ozempic and Wegovy were made. But you see all the biosimilars are being made through a process called SPPS, which is solid phase peptide synthesis. Tirzepatide is made from solid phase peptide synthesis. So is Mounjaro a bad molecule? So this argument of that we have made a biological molecule and they have made a synthetic molecule doesn't hold waters. It is not actually correct. This argument doesn't hold at all. But having said that, we need to understand that the two processes of making the molecules are different. And because they're different, they accumulate different impurities. And that is what all the authorities want to see. What is the impurity that you are accumulating? Because the final product must have more than 99% purity. This is the recommendation from all the bodies. 98.9 won't do, 99% purity won't do. It has to be more than 99%. So this is Indian regulatory body. Nothing is moving actually, so I'm saying up here. Let me see. So this is the Indian regulatory body, CDSCO's guideline. You see the most important is analytical similarity. The primary structure, secondary structure, and tertiary structure has to be the same. So 3 questions we need to ask. Second is the PK, pharmacokinetics. The maximum concentration that the drug reaches must be the same, and the disappearance from the system must be exactly at the same time, and impurity. Clinical part is optional, it is not mandatory. Now, I have this data, I'll not show, so I've written A and B from 2 different biosimilar companies who agreed to share certain portion of the data with me on their laptop. But somehow I found it in some public forums, so I'll be showing what they have done. Now, of the 2 companies, one of them have showed 0 anti-drug antibody, which is nearly impossible. There has to be some degree of impurity. They've given you 80 to 125% variation, which is allowed. 0. And what they found is they have not given the time when the drug disappears from your system. The data is not given to us. No statistical analytical framework of the analytical study, primary, secondary, tertiary structure was given to us. Unexplained superiority signal. When the study was done, the A1C in the biosimilar arm was superior compared to the other arm. Now that is strange. That doesn't make it the same molecule, right? The other one had showed perfect HbA1c and weight equivalence. In the equivalent study, the A1C reduction was 2.22 and biosimilar arm was 2.22. The weight reduction in this arm was 6.5 kgs, the weight reduction of other was 6.5 kgs. We have never seen such similarity ever in any studies that we have done. This is absolute, matching the data to data. How that has happened, we don't know. Broad scope detailed data set, there's a lack of batch-to-batch reproducibility. neutralizing antibody is not required by the Indian authorities. And if you have anti-drug antibody, which is 0, so what is the role of a neutralizing antibody? So these questions are being asked, but these are probably redundant. So this is what our regulatory body, and this is what FDA wants. There are a few gaps in between, which needs to be fulfilled. So whenever a company says we have got the Indian approval, let's wait what happens when it comes to an international approval. One molecule we need to be extremely careful about is the oral semaglutide. The oral semaglutide is not oral ribelsus. It's a completely different molecule. So if you think that I'm going to make my oral ribelsus cheap by writing this oral semaglutide, you're making a big mistake. They are not the same molecule. It falls under section 505B, subsection 2, as follow-on biologics. Means they need to do the clinical trials again. They need to do a trial like FLOW and others once again to claim that they are equivalent to the other. Why is that? Because the original molecule has a transcellular absorption. As I told you, to be biosimilar, you need analytical similarity, and 2, you need absorption similarity. But the new molecule, the absorption is both intracellular as well as paracellular. It's a completely different absorption pattern. So they are 2 different molecules. It's something like you have a semaglutide molecule with something else, A attached to it, and a semaglutide molecule with B attached to it. A plus B is not equal to A plus C. They are 2 different molecules. So you cannot extrapolate at all. The injectables, you can, but the orals, you cannot. So semaglutide plus SNAC is not equal to semaglutide plus the dual PE system. And hence, all the cardiovascular and all benefits that we give our patients when we give them an oral semaglutide cannot be extrapolated to the present day oral, the generic that we have got in the form of oral semaglutide. So this is something we need to be extremely careful. So if my take is, if my patient has a compelling indication, why am I giving you my final slide, which is this, is if you see the indication given to the biosimilar semaglutide are only for management of type 2 diabetic and for obesity. There is no indication for CVD, for MASLD, for CKD, etc., etc. So if your patient has these problems, it is probably better now, even now, to give them the original molecule. Thankfully, by competition, even they have cut down the cost. So even their cost has come down. But if you have a simple type 2 diabetic patient, simple obesity-related patient, and genuinely the patient can't afford, we can definitely give them the biosimilar semaglutide that we have. So we need to understand a few things. Different regulatory bodies have different rules. So we have got approval in India, but I'll show you 2 important slides. Dr. Reddy's went for an approval in October 2025 in Canada, got rejected. It is not accepted, and they have to refile for the application. 6 days back, Brazil has rejected both Cipla as well as Reddy's biosimilar semaglutide. So these are the markets who have rejected the molecules because of the analytical structure problem. They have not given adequate data. Probably they will come back with a fresh submission, and they will get accepted in the near future. As of now, they're getting rejected in every international market that they're trying to enter. So we need to be a little bit careful. High-risk patients, we prescribe evidence. In low-risk patients, we can prescribe equivalence. And in real world, cost determines whether treatment happens at all. So having said all those things, I'm not scaring you. Write the appropriate molecule. Please be careful, because there is no standardization on the type of pens being used. This is a 100% recommendation in the international bodies. The similarity also has to be the pen. Now I have 2 pens here. I'll not tell you which company. But if you see in one of the pen, there is a huge gap. The piston is here. You have to 5 times click it, which is blank shot, for this to come near the piston. And from the 6th shot, the patient will get the injection. This is an absolutely defective pen. You cannot have a pen like this. This is an absolutely beautiful biosimilar pen. Everything is in place. So whenever you choose a biosimilar for a patient who can't afford, please check the pens yourself. Some companies are selling pens for 6 months when they themselves have done the study where they change the cartridges every 6 weeks. So you have done a study by changing cartridge every 6 weeks, and to make it less expensive, you're giving a pen lasting for 6 months. My patient develops GI side effects, discontinues after 2 shots. You are actually still wasting money and a lot of drug. So it doesn't make sense. So please check pens. We have very good biosimilar pens also available. Appropriate pens, doses is beautiful. Like Trulicity, we have single shot pens also available. So be a bit careful. Ask the appropriate question to the companies when they come with the pen and check the pen. These are the two very important things that we should consider. Thank you.